Okay, good afternoon, everybody. I'm really glad that you guys could all make it to our colloquium today. Hopefully you all enjoyed your spring break, so it's nice to see you all here again on a Monday, right? Well, not too early, but ready to go for sure. Today our honored guest is going to be Dr. Connie Mulligan, who is at the University of Florida. She is a professor in the Department of Anthropology and the assistant director of the University of Florida's Genetics Institute. She has a variety of research interests, but at the most fundamental level, she is interested in understanding the basis of patterns and levels of human genetic variation. So her biocultural investigations on human disease have engaged many issues, and in the past her work has looked at the peopling of the New World, domestication of animals, and genotype-phenotype associations with various disorders. And what she'll be presenting today deals with her interest in that intersection between genetic, epigenetic, biological, psychosocial, sociocultural impacts on complex human phenotypes. The talk is titled Genetic, Epigenetic, and Sociocultural Data as Applied to Questions of Human Health and Disease, in which she'll be describing two of her projects in her lab, one which examines the genetic and cultural risk of factors for hypertension in African Americans living in Tallahassee, Florida, and the other of which investigates an epigenetic mechanism to mediate the effect of maternal stress on maternal and infant health in the Democratic Republic of Congo. So please join me in welcoming Dr. Connie Mulligan.
Thanks so much. It's a real pleasure to be here. It's always nice when the grad students in particular invite you. So I'm quite honored to be here. So like Tiffany said, I'm going to be talking about genetic, epigenetic, sociocultural, biological, psychological effects on different complex phenotypes. So I'm interested in complex phenotypes, which are defined as those that have multiple genetic and multiple environmental effects. And once you sort of open the can of worms of environmental effects, you know that it's sort of no holds barred. So that's why I say sociocultural, psychological, sort of anything that impacts complex phenotypes. And I'm particularly interested in human complex diseases, again, those with multiple genetic and multiple environmental effects. And I'm more particularly interested in those that have a stress component, because stress is so prevalent in our society, and also stress has multiple facets: genetic, biological, sociocultural, psychological. I'm also fascinated by the idea that epigenetic mechanisms might actually mediate the effects of stressors in one generation, possibly in a multi-generational way, where effects from one generation may be seen through multiple generations. I'm also very interested in racial health disparities, again because they're so prevalent in our society, and again because they have multiple genetic and environmental causes. So I'm most particularly interested in studying complex human diseases that show racial disparities, because I truly believe that by taking a comprehensive approach that takes into account genetic, biological, sociocultural effects, that we could get a better handle at the origin, diagnoses, treatments of these different diseases. And I think that an anthropology department is the perfect place to take this very comprehensive view. And so I feel very fortunate in being both in the anthropology department and affiliated with the Genetics Institute, where I get to do this research that I think is fascinating and also a lot of fun. So my research is inherently interdisciplinary, and so with interdisciplinary research you often have different fields converging on the same issue or same concept, and they all bring their own terminology. So all of these terms here are an example of that, in that all of these fields or approaches, molecular anthropology, human sociogenomics, social genomics, biocultural approach, social and behavioral epigenetics, are all essentially saying the same thing, where we're looking at both molecular genetic data and then, from my perspective, since I'm a geneticist, I sort of lump everything else into non-molecular genetic data to get the most comprehensive view on what's going on. And as my research has developed, I've become very aware of the different types of data that I'm incorporating and how they're collected in different ways. They're analyzed in different ways. From a certain perspective, they're very different kinds of animals. And so I want to talk just a little bit about that. So I started as a geneticist, and as I started to look at different types of data, it struck me how simple DNA data are. You have 4 variable sites. You've got A, G, C, and T. We can sequence someone's entire mitochondrial or nuclear genome. We can sequence it to hundreds X coverage. We can calculate the variation associated with any site. We can then sequence multiple individuals. We can calculate diversity levels across populations, across genomes. We can then calculate the variance associated with those diversity levels. Another aspect of the relative simplicity of DNA data is that DNA data sets are easy to standardize. So almost all of these data are then put in public archives so that other people can download our data and can double check our diversity measures, double check our conclusions. And then finally, with the advent of next generation sequencing, we often don't even have to decide which gene or which single nucleotide polymorphism or SNP we want to analyze, because we can kind of do it all. We can just look at the whole genome and then just correct for multiple testing. So then I moved into epigenetic data. And right away there, and when I talk about epigenetic data for today, I'm really going to confine my discussion to the methylation of CpG sites in the human genome. But it immediately became different that epigenetic data are different than genetic data, because as opposed to having binary sites where you're an A or a T, you're an insertion or not, you can have levels of methylation that range from 0% to 100%. So the difference is between a light switch versus a dimmer switch. And epigenetics is a relatively new field if we don't think about imprinting diseases. So epigenetics more at the social and behavioral level is a new field. And so there have been publications talking about how we ought to standardize the data and guidelines as to how to interpret the data. And while I'm all for guidelines so that we're all comparing the same thing, I would also caution against too much of an over-reliance on hypothesis testing, because social and behavioral epigenetics is a really new field. And when you define a hypothesis, it almost by definition is very narrowly focused, so as to be falsifiable. And in that narrow focus, then you're kind of ignoring other things. You're either purposely or maybe not purposely, not aware of the things you're ignoring, but you're making a conscious decision to formulate this rather narrowly focused hypothesis. And I think that with methylation data, we're not really at the point of knowing what to ignore. We still have some very basic questions we're trying to answer. So in general, if a CpG site is methylated, if multiple CpG sites are methylated, it's correlated with a reduction in gene expression. We know that's generally true. There are exceptions. But the devil's in the details. So how many CpG sites have to be methylated? Is there a critical number that have to be methylated? Are there key sites that have to be methylated? Is there some critical level of methylation that must be reached before you get a change in gene expression? I'm particularly interested in the way in which environmental stressors might change methylation marks and then ultimately change gene expression. So when we talk about environmental stressors, so the first question is, do environmental stressors change methylation marks in any way? But then the next question is the persistence. How long might that change in methylation marks persist? Through an individual's lifetime? Through multiple generations? These are all really fascinating questions, and they're questions that we don't know the answer to. So I think, so the way I phrase it is that we're not hypothesis testing, we're hypothesis developing. So that's sort of my, nicer phrase than fishing expedition. All right, so now we move to cultural data. So here it's a lot more complicated. Cultural factors can include diet, physical activity, stress, environmental toxins, socioeconomic status, education, psychiatric diagnoses, the neighborhood in which you live, pretty much everything, including the kitchen sink. And these data are collected in a completely different way than genetic data, often by interviewing a study participant. So now all of a sudden you're completely dependent on the understanding, the commitment, the personality, the self-awareness, the memory of your study participant in order to guarantee the accuracy of your data. You also have to consider that some study participants will give you the answers that they think you want to hear, as opposed to the answers that are really true. There are things that we can do to improve the accuracy of these data, but at the end of the day, you just don't have as much control over these data as I like to think I do over my DNA data. And so not only that, but you have to make some hard choices about what questions to ask, what data to collect, because you can only have a questionnaire that is so long, or you're going to get into issues of interviewing fatigue. And so we've had to grapple with that, because already some of our interviews are 2, 3, 4 hours long, or the open-ended interviews have gone on for even longer than that. So you have to make some hard choices about what data you are going to collect and what data you have decided not to collect. All right, so with that as an introduction, I'd like to talk about 2 of the projects ongoing in my lab that I'm most excited about. And I'm going to start with our research in the Democratic Republic of Congo. And here, I'm particularly interested in the idea of looking at the possibility of an epigenetic mechanism that mediates stress to the mother into changes in the mother's health and also in the health of her newborn. And so that gets at this idea of multi-generational effects of stress. So I'm really intrigued by the idea of this 9-month period in which the mother and fetus share an environment, and in which the fetus is subject to all the stressors that the mother experiences. And I don't include just the stressors that she directly experiences during her pregnancy, but also stressors previously experienced that may or may not still be having an impact during that 9-month period. So I'm interested in how maternal stress then affects the mother's health and also her baby's health. And this is a collaboration with former grad student Nikki Rodney, who actually started this work in Congo. Darlene Curtis is a professor at UF in the Department of Psychology. And then David Hughes is a former postdoc, and Jackie Quinlan is a current postdoc, who are all working with me on this project. So we all experience stress. We all know stress is bad. We know it's bad for our health, for our happiness, for our job performance, but we really don't understand how. And to make matters even worse, what if stress possibly can affect next generations? So I'm really interested in the way that this might happen. So at a very simplistic level, we know that if a pregnant woman doesn't get proper nutrition, her infant is affected. And there we can pretty easily understand how this happens. But what if it's more subtle, more nuanced than that there are psychosocial stressors? And again, we know that the infant is affected, but exactly what sort of mechanism, how is that information about the maternal stress transmitted to the fetus, and particularly transmitted in a way that might affect disease risk in that infant decades down the line? So epigenetics seems to be a perfect sort of candidate as a mechanism to convey, to transmit this information. And that's what really fascinates me about this project. So this is the model we set up. We've got this proposed relationship of environmental stress influencing some kind of outcome phenotype being mediated by methylation that then presumably has an effect on gene expression. So like I said, I'm interested in the effect of stress on maternal and infant health, particularly interested in the heritable effects and the idea that the infant is exposed to maternal stresses through that 9-month period. The developmental origins of health and disease hypothesis, or DOAD, posits that early life experiences are related to disease risk in later life, and particularly posits that there's a critical period of developmental plasticity in utero, and that it is during this time period that the range of possibly infinite phenotypes is narrowed down to fewer and fewer, possibly a single phenotype that is chosen based on the cues from the uterine environment and based on what should be most adaptive given that environment. But if that selected phenotype is not the right one, or if the subsets chosen are not the right ones, it may actually be maladaptive in later life and may explain increased risk of certain certain diseases decades later after birth. So we work in Congo, specifically Eastern Congo, which unfortunately is always in the news. This quote here from 2012, Congo has been subject to ongoing civil war and constant violence against women, making it one of the most stressful places in the world. So even though we know that stress has an effect on health, I argued, or I thought, that if we're really looking for an effect of stress on the genome, on the epigenome, it's not going to just jump out and tell us here it is, particularly since we don't know where to look, so we're having to look across the entire genome. So I argued that we needed a really extreme stress phenotype to try and figure out what parts of the genome or epigenome were being affected. And unfortunately, Goma, where we worked, provides that very stressful environment. But so even though we're arguing that we need this extreme stressor phenotype to see an effect on the genome, I do believe that the results we find will then be relevant to just everyday stress once we figure out exactly what parts of the genome or how the genome is being affected. And furthermore, even though we'd like to say that a lot of what goes on in Congo, particularly the sexual violence against women, is, limited to over there, we know it's not. There was a recent study showing that 1 in 5 women on U.S. college campuses will be raped during their time in college. So unfortunately, I'd argue that all of our results are directly applicable to U.S. populations. So like I said, I worked with Nikki Rodney, who started this project, and it's no exaggeration to say she took her life in her hands collecting these data and samples. Nikki's absolutely passionate about maternal and infant health. The women that she worked with were equally passionate about her. Many of them named their babies after her. They told her that God had sent her to them to tell her story, and that's what we're trying to do. So Nikki collected from 25 mother-infant dyads, venous blood representing the mother, placental tissue, and then umbilical cord representing the newborn, and then also did extensive ethnographic interviews and trauma surveys to develop EMIT measures of maternal stress. So this was all done in 2010. This is sort of our pilot study with 25 mother-infant dyads, and we're now into the second stage where we've got 100 mother-infant dyads, but most of what I'm going to present today are on those initial 25 mother-infant dyads. So we first took a genome approach. This was before I had a grant, so I was doing it on fumes of funding. So you can't look at the whole genome, you look at a single gene. We decided to look at the glucocorticoid receptor NR3C1. It was an obvious first place to start because NR3C1 has been associated with newborn birth weight for many years, and more recently, altered methylation in gene expression at NR3C1 has been associated with childhood abuse ending in suicide, PTSD, prenatal exposure to intimate partner violence, and it's also been related to predicting treatment outcome in PTSD veterans. So it was sort of the obvious first gene to look at. And then relating it back to our model, we wanted to say, does maternal stress influence methylation at influence our outcome phenotype, which was newborn birth weight, via some sort of altered methylation pattern at NR3C1, specifically the upstream region of NR3C1, which has 39 CPG sites that we assayed for, and all those other studies I told you about, that's the same region they assayed for, so there's nice overlap between the studies. All right, so these are the questions that Nikki asked. We developed based on these questions, we then developed 3 additive models of stress. Material deprivation to reflect the availability of financial resources. Do you own your own home? Do you have a private room in your home? Mundane stress to reflect daily psychosocial stress. Did you have any prenatal care? Were you stressed during your pregnancy? Do you have a happy marriage? And then more stress to reflect some of the war-related events. So have you ever been raped? Is your current pregnancy a result of rape? Have you had a previous pregnancy a result of rape? Have you seen a family member killed? And unfortunately, even in our very small sample size of 25, we had multiple yeses to all of these questions. So the first thing we wanted to look at was the effect of maternal stress on birth weight. And we looked at our 3 different stressors, and we saw strong correlation with each of those 3 stressors in birth weight. In other words, increased stress was correlated with decreased birth weight. We saw the strongest effect with war stress. In fact, war stress accounted for 35% of the variance in birth weight. And if we look just at personal experience of rape, those questions accounted for 31% of the variance in newborn birth weight. So that's a really big amount of explanatory power for a complex phenotype like newborn birth weight. So we really thought this meant we were onto something here. So, but going back to our research model, we've now shown this effect of maternal stress on our outcome phenotype of newborn birth weight, and this then serves as a foundation against which to then test for an epigenetic effect or pushing even further an epigenetic mediation. So when we looked at the results of sequencing across the different methylated sites in our moms and babies, the first thing we saw was that there were no identically methylated sequences between any mom and her child. So that was kind of surprising. We interpreted that to mean that there is possibly very little genetic influence on methylation of this upstream region of NR3C1, and possibly then opens it up for a potential of greater environmental influence on the methylation at this upstream region of NR3C1, suggesting possibly that NR3C1 is a gene that is more able to evolve and adapt in response to environmental stimuli. All right, so this is now looking at for a correlation of methylation with our stressor and with our outcome phenotype, war stress and birth weight. So on the top of the graph, we've got newborn methylation. On the bottom of the graph, we have maternal methylation. So we only see a significant correlation with newborn methylation. We see a significant correlation between maternal war stress and newborn methylation. This is the first component of a principal component analysis of the newborn methylation. And we also see a significant correlation between newborn birth weight and newborn methylation, but no significant correlation with maternal methylation. So here our association is increased stress is associated with increased newborn methylation, which is then associated with decreased newborn birth weight. So we've now completed the model where we've got the correlation of stress with newborn methylation and newborn birth weight, which completes the cycle. So a correlation with newborn methylation only is consistent with the tenets of the DOAD hypothesis that suggests that the newborn is in a particularly plastic state as opposed to a full-grown adult or mother. And that as a result of being in that state is more able to choose and discard different phenotypes based on interpreting intrauterine cues. And so this is how we get into the idea that early life experiences could be related to disease risk in later life, particularly in the infant, who's the only one we're seeing the correlation with the methylation. And that NR3C1 may be one of possibly a very small handful of genes that are able to mediate the effects of the environment. In other words, we don't expect there to be a ton of genes or a whole genome-wide effect to mediate the specific tenets of the DOAD hypothesis. Okay, so how might this correlation work? So like I said before, the idea is that a stressful intrauterine environment may select for a phenotype that is ultimately maladaptive. So how could it be maladaptive? So increased methylation at NR3C1 predicts that there would be decreased gene expression of this gene. So it's possible that this increased methylation will then restrict the different responses of gene expression at NR3C1 because it's just starting out at too low a level of gene expression. And we know that if there's an imbalance in the glucocorticoid and mineralocorticoid receptors, that this increases the risk of stress-related psychiatric disorders. So if the increase in methylation and presumed decrease in gene expression we see at NR3C1 isn't matched by a similar response in the mineralocorticoid receptors, we would then predict that this individual would be at risk for later life stress-related psychiatric disorders. And then the fact that we did get a correlation with all 3 of our stress measures, we just got the strongest correlation with the war stress, but we got correlation with all 3 stress measures, has implications for the growing prevalence of stress disorders in the U.S. All right, so now we move, so then I got a grant, so we could spend more money. So we went to a genomic approach. We used the Illumina 450K bead chip to look at about 450,000 CPG sites across the human genome. And for these results, I'm really just going to present the war stress results because, again, they were consistently stronger. All right, so here we're looking for a correlation between war stress, and now that we're looking genome-wide, we've got a genome-wide mean methylation measure that we're using to encompass all of our methylation data. So we're looking for a correlation between war stress, genome-wide mean methylation, and birth weight across our three tissues, so placenta, cord blood or infant, and then maternal venous blood. And here we see that we only get a significant correlation between venous methylation and war stress, venous methylation and birth weight. So we only see a correlation with the mom's methylation, which contrasts the results I just showed you with NR3C1, where we only saw a correlation with the newborn. So first of all, we were really happy to see a significant result at all because we honestly didn't know if this mean methylation across the genome would be a sufficiently sensitive measure to pick up the effects of stress. I mean, so to some extent it's saying that stress is impacting across the genome, which is kind of scary. But the idea that we only find it in the moms makes sense in that the individual directly experiencing the stressors, that is the individual we would predict to show the biggest effect, particularly on a genome-wide level, and that is indeed what we see. So the fact that it's different than the NR3C1 results might just mean that different things are going on at a genome-wide level than are going on at a specific gene level. So, getting back to our model, so we have an effect of maternal stress, change in maternal genome-wide methylation, still correlating with our outcome health phenotype. Here we've got increased maternal stress is correlating with decreased maternal methylation, which correlates with decreased newborn birth weight. So just as a quick aside, some of the people at UF who study imprinting and so particularly study methylation at a molecular level are somewhat suspicious of a lot of the social and behavioral epigenetic data that are being produced because there's not a lot of de novo methylation that happens after you're born. So they said a signal of decreased methylation is actually a lot more explainable than if someone had a signal of increased methylation. In other words, there are mechanisms, there are enzymes present in humans that can account for losing methylation. But to create methylation after you've been born is a lot harder because there simply aren't the enzymes to do it. Whereas in utero, there are enzymes that are methylating. I mean, that's part of what happens during pregnancy, during development. Okay, so the idea here is that the individual most directly experiencing the stressors we might predict would be the one most likely to show a genome-wide effect. We don't see it in the infant. It might be because they haven't developed yet. We've only got cord blood to reflect the infant. It might be that the changes to the infant, particularly consistent with the DOAD hypothesis, would be present only in a select subset of genes, those that have possibly evolved to be more sensitive to environmental effects. All right, so this is a summary of our two sets of results. I've pretty much gone over all of these. So genome-wide is manifested by the individual most directly experiencing the stress, and if we're thinking DOAD, at least by my way of thinking, if we look across the genome and we look at genes and we look at what drives their methylation, it seems like we're going to have some sort of housekeeping genes whose methylation is just genetically determined. We really don't want the expression of those genes changing if I'm stressed or if I don't have a good breakfast or something like that. We want those genes to do the same thing, make the same level of protein day in and day out. Then on the other end of the spectrum, there might be some genes that have evolved, particularly in higher order organisms like humans, to respond to the environment, to allow us to adapt faster than our DNA sequence can adapt. But it makes sense that it would be just a small subset of genes because we don't want a lot of our genes going off and, changing their expression by kind of signals that can be very transient. So that's where we are with these data. We then did a couple other things with it. So we pooled all the tissues just to look across all tissues as a way of controlling for cellular heterogeneity, and then looked across all tissues to see if there was a correlation of war, stress, and methylation. And at a 5% false discovery rate, we found 212 CPG sites, 13 promoters, and 37 genes out of these that we started with. And what's more interesting is that then David derived a sort of calculation for the heritability of methylation. And of these sites that he found to be significantly associated with stress, 43% of them were also observed to be heritable. So this is then consistent with the idea that CPG sites that are correlated with maternal stress are also largely identified as heritable. So then the last thing we looked at with these data was in a genomic context. Where should we be looking? Should we be looking at gene bodies? Should we be looking at introns? Should we be looking at promoters or enhancers? A lot of focus right now is on promoters. So the Illumina 450K bead chip is heavily dense with CPG sites and promoters. But what you see from these two graphs, so this is a graph looking at all three of our tissues. This graph looks only at the two blood tissues, so the cord blood and the venous blood. And then on the y-axis, we've just got mean methylation levels, and on the x-axis, we've got mean differentiation of methylation across 3 or 2 tissues. And so what you see down here are promoters and actively expressed promoters, but what you see is that they have the lowest levels of methylation and the lowest differential methylation across tissues. Whereas up here is where enhancers are. Enhancers have both the highest level of methylation and the highest differentiation of methylation across 3 or 2 tissues. So this is looking at differentiation across different tissues, but it stands to reason that enhancers might also be the place to look at for different levels of methylation in response to environmental stressors. So these results suggest that methylation variation in regulatory regions outside of promoters might actually be the place to be looking for a role in regulating tissue specificity, but then also response to environmental stressors. So let's just take a quick look, closer look at our stress questions. So this was the first way we analyzed the questions. And we put them in these categories: material deprivation, mundane stressors, and war stressors, because they kind of made sense. We got a nice correlation with methylation and birth weight. But it wasn't necessarily the only or best way to do it. So Darlene Curtis, my collaborator in psychology, did a factor analysis on these same data and came up with that 2 factors actually were sufficient, and in fact did a better job of accounting for all the variation in those questions. And so if we take a closer look at these 2 factors, component 1 explains about half of the variance, component 2 explains about 9%, really high internal consistency within both of these factors. Component 1 seems to largely reflect chronic stress, and component 2 more reflects war and acute stress. We've got 3 stressors that load on both components. Have you been raped in the past? Are you afraid at night? And did you decide the birth location of your current pregnancy? Meaning that these, in fact, might be stressors that have both chronic and acute components to them. And then, interestingly, component 2 is mainly composed of our old war stressor measure, with 2 exceptions. Have you been raped in the past? And have you had a child of a past rape? These are now loading on the chronic stress component, arguing that those events, they have turned into chronic stressors because of the nature of that stress. So what this means to me is that we really do so it's good to take the statistical approach to be able to say we can support these measures statistically, but also to look at the different questions and how they're loading and how the analysis suggests that these stressors are then being experienced by the women. So we're now going forward with these stressors, and Darlene just had a paper accepted in a special volume of child development showing a correlation between these stressors and different genes in the HPA axis. And we're just carrying on with these stressors now for additional analyses, but now we're moving on to the set of 100 diets. Okay, so our conclusions. So we think that stress may induce both a genic and a genome-wide effect on methylation that may be heritable. We see the newborn change in NR3C1 methylation, and then maternal genome-wide changes that correlate with maternal stress. We see a high heritability of the CPG sites whose methylation is correlated with stress, suggesting that this effect of stress may be a heritable change in DNA in methylation. And we also suggest that non-promoter regulatory regions like enhancers may actually have a larger role in methylation that is variable tissue to tissue or by environmental stressors. So for the future, like I said, we're actually well into the second collection phase of 100 mother-infant dyads. I got much more ambitious. I had a very long wish list for these samples. So we got blood from everybody, enough for 2 samples of DNA and 2 of RNA. We got from the mother, placenta, cord blood. We got baby at birth and baby at day 1. We got extensive medical histories on both the mom and baby. We did the stressor questionnaire with the mother. We also collected saliva, 3 samples for cortisol testing from the mom for diurnal cycling and from the baby to look at stress reactivity. We got hair samples from the baby to look at long-term measures of cortisol. We also got dried blood spot samples from the mom and baby to look at biomarkers. And then since it was going so well, better than I ever could have imagined, we then thought, well, let's go for follow-up samples. So we've now got 1-month follow-ups from just a subset. Again, we got all the same samples that we'd gotten from baby. From mom, we did the stressors checklist, but we also started using different questionnaires to look at depression and anxiety. We've moved on to 6 months, and we've got some 1-year-old samples, and we're now in the planning stage to do 2 years. So Jackie's going over to Congo in about 2 weeks to explain to the moms who don't understand why we need them to keep coming back and giving more blood sample, just to explain that, you know, it really is a long-term sort of project where we want to follow the health and then cognitive development of the babies as they age, and then continue to collect samples along the way. And getting even more ambitious, it'd be great to have samples from the dad, samples from the mom's parents. We're also looking into ways in the field of separating out cell types so that we can take care of cellular heterogeneity. We also want to start collecting blood so that we can look at proteins to fill in that third step in our model. So we got stressor, we got methylation, we got the outcome phenotype, but in the middle there was gene expression. So we'd like to be able to look at proteins as well. So I figured this will keep me busy for a while. Just to end the Congo project, I'd like to go ahead and thank all of the women who have participated in our study. They've been great. Our team in Congo who collects the samples are amazing. I would not have thought it possible to have a project this complicated with this many different types of samples and data being collected, and everybody collecting it is our Congolese collaborators, and they're awesome. And Felicien Masango, who is the leader of the project, will be coming over to the department in fall to do his PhD in our department. So I'm really hopeful that it means the project really has long legs and will continue well into the future. So the next project I want to talk about is our study of genetic and sociocultural risk factors for hypertension. So we're interested in studying how genetic, biological, sociocultural factors contribute to influence health. And we're looking at blood pressure. Blood pressure is great to study because it's relatively easy to measure, yet it's a complex phenotype, again, with multiple genetic and environmental factors. This project's a collaboration with Lance Gravely, who's a cultural anthropologist in my department, and then Laurel Pearson, a former postdoc, and then most of the data I'm going to show today are hot off the presses by current postdoc Jackie Quinlan, grad student Chris Kluke, and undergrad Alexis Bolter. So our project uses hypertension as a vehicle to investigate the different factors that influence blood pressure, and also because hypertension is a risk factor for cardiovascular disease, ischemic heart failure, and chronic renal failure. So hypertension affects over 70 million Americans and is a major public health problem. But we also study hypertension to investigate racial disparities in health. So NIH defines health disparities as difference in incidence, prevalence, mortality, and burden of diseases and other adverse health conditions that exist among specific population groups. So data from the National Center for Health Statistics in this figure show that black men and women have shorter life expectancies than their white counterpart, and black men in particular have the shortest life expectancy at all. So in the U.S., for every major cause of death, the burden of poor health is patterned along racial lines, and the burden's especially heavy for blacks who experience higher rates for most leading causes, including heart disease, stroke, cancer, diabetes, influenza and pneumonia, and HIV/AIDS. So as a result, life expectancy at birth remains about 6 years shorter for black Americans than for white Americans. So then bringing it back to our study, blacks are disproportionately affected by hypertension, and specifically with cardiovascular disease, in comparison with non-Hispanic white Americans, and have a 30% higher instance of associated mortality. So the question we're particularly interested in are how do genetic and sociocultural risk factors affect risk of hypertension, or actually what we measure as variation in blood pressure. And we're particularly interested in the effects of discrimination on blood pressure. So we've identified factors that we think are most important in influencing blood pressure, such as genetic data that capture candidate genes, and also genetic data to estimate genetic ancestry, and then also cultural data on an individual's race, their socioeconomic status, their experience with discrimination, and a whole lot of other measures that I'm not going to have time to talk about today. So we work in Tallahassee. We're looking at hypertension in African Americans living in Tallahassee. Tallahassee has a large black middle class because it is the seat of the state government, and also Florida Agricultural and Mechanical University, which is one of the largest historically black universities. There is a very large percentage of African Americans who hold graduate degrees, who have high socioeconomic status, but there's also a quarter of African Americans who live below the poverty line in Tallahassee. So it means in Tallahassee we have a very broad range of socioeconomically and educationally diverse African Americans. There's also a long history of racial struggle and civil rights activism in Tallahassee, and individuals living in Tallahassee have identified racial health disparities as a particular problem that they want to address. So we take a community-based participatory research approach, or CBPR, in Tallahassee, where there's a lot of engagement with the local community, and particularly we have a very active steering committee composed of all local people. One's a retired pediatrician, one works for the State Department of Ministry, one is sort of a neighborhood organizer. One, Maisha, is the backbone of everything. She runs one of the biggest sort of programs in Frenchtown in Tallahassee, and they were involved in all aspects of the study, from designing our very non-standard informed consent form, suggestions on how to tell people about the study, how to recruit study participants, how to report results back to them. We had a meeting with them just last month talking about just moving forward. So in 2011 and 2012, Lance Gravely collected all the samples that my lab has analyzed, but also just a wealth of sociocultural data. And mostly what I'm going to be talking about today are data that have to do with discrimination. So he developed some new measures of unfair treatment and everyday discrimination. He also collected a lot of data on neighborhood stressors. Do you feel your neighborhood is safe? Are you able to, do you have access to healthy food in your neighborhood? And then also, he's looking at personal social networks. So this is specifically to test a hypothesis that came out of his graduate work. And the hypothesis is that darker-skinned individuals at high SES levels experience increased levels of discrimination relative to darker-skinned individuals at low SES levels because, presumably, of their work environment. So we're going to use personal social networks to actually quantitatively test the hypothesis if that is indeed true. So then my part of it is the genotyping of all the samples Lance collected. We ended up with 170 saliva samples, and then Laurel Pearson, a former postdoc in my lab, designed a custom Affymetrix microarray that had about 250 genes or 25,000 SNPs involved in hypertension, cardiovascular disease, pigmentation, stress, methylation, and taste. and then about 5,000 SNPs to estimate genetic ancestry. And then we also looked at some non-SNP alloinsertion deletions that had been previously identified in the literature as important. We're also very interested in adding an epigenetics component to this project, and we also have dried blood spots that we can look at biomarkers of stress, or biomarkers that reflect general health indices. So the three questions that I'm going to talk about today. Is there an association between blood pressure and genetic loci or candidate SNPs? Second, does genetic ancestry or admixture mapping strengthen the analysis? And finally, what happens when we add in some of our measures of unfair treatment? So Jackie started looking at different ways of analyzing data like ours. So like I said, it's a custom-designed Affymatrix chip that has about 250 genes on it, about 25,000 SNPs. So it's kind of more than a genic approach, but kind of less than a genome-wide approach. It's somewhere in between, right? So you could kind of do association mapping on it, where you're looking to detect associations between particular SNPs and your phenotype. We could also do admixture mapping on it, because we have ancestry informative markers, which are needed to estimate genetic ancestry. So she found a paper by Schreiner et al. That actually combines the two methods, integrates admixture mapping with standard genome-wide association studies. And Schreiner argues that this method is particularly good for detecting signals in admixed populations, like we have, and for detecting signals where you've got SNPs that lie in a region where you might have multiple independent causal variants. And so leveraging the information from those potentially causal variants that are lying in the same region is basically what this paper does. So Jackie went ahead and did this analysis. So this first slide shows a typical admixture mapping. So across all the chromosomes are 25,000 SNPs, then looking at systolic blood pressure and diastolic blood pressure, and then correcting for multiple comparisons. In this case, she used a strict Bonferroni correction, and she corrected for the number of blocks of SNPs, of ancestry blocks that were identified, or the number of times that a block switched. It's just called haplotype switching. So there were 80 switches that she then corrected for. So then, given that Bonferroni corrected p-value, none of the SNPs were significantly associated with blood pressure. Then she did the sort of standard genome-wide association analysis, and she got two SNPs. They're so close together they appear as a single dot here. So here she did, again, strict Bonferroni correction for all 19,000 SNPs were the ones who made the final cut in terms of data quality. So up to 2 SNPs. Then she combined both, the ancestry and the association, and combined both correction factors, so both the 19,000 SNPs and the 80 haplogroup blocks or switches. And now she came up with a couple dozen SNPs that's significantly associated with systolic and diastolic blood pressure. So that's sort of a number that, is nice to work with. So if we look at, so the first thing you want to do is, well, what genes? Where are they occurring? So, we've got this list of SNPs. We've got, where they occur in a genome context perspective. But how do you visualize all of this? So the next thing Jackie did was add in these new measures of unfair treatment, since that's really what we're interested in. I'm really not interested in doing another association analysis that finds new SNPs associated with blood pressure. My interest is really looking for interactions between the genes and novel sociocultural data, and how that might give us a better understanding of what's going on here. So like I said, Lance Gravely developed some new measures of unfair treatment. So he took existing questionnaires and then just kind of modified them based on his ethnographic interviewing. And so one thing that came out of his ethnographic interviewing was that when people were given an open-ended question, talk to me about discrimination, oftentimes they wouldn't talk about themselves. They would talk about people close to them. And this is something that no existing questionnaire on discrimination taps into. The idea that discrimination that happens to someone close to you might be as salient as discrimination that happens to you personally. So he modified the existing questionnaire with the underlined words. So the question would read, have you or someone close to you been unfairly treated by the police, been unfairly treated by the courts, been unfairly treated at school, in getting housing, in getting money or a loan. So these are the sorts of questions. There were 9 questions, and so Lance's only modification was to add in these words, or someone close to you. Then if someone answered yes to any of these questions, the interviewer circled back and said, you said yes. Was it you, your spouse, a parent, a child, a grandchild, another close relative, or a friend, so that we could track who these others were. Then there was a separate measure of everyday discrimination, again using an existing questionnaire that Lance then slightly modified. And the questions here were, how often are you insulted or called names? How often are you threatened or harassed? How often do people act as if they're afraid of you? So these 2 measures of the 2 questionnaires were originally developed to reflect acute versus chronic stress. I don't necessarily agree that this is more acute and this is more chronic, and I'm definitely not going to try and defend that. But what I take out of this is that there was some thinking that these 2 types of stressors might somehow be different, possibly different biologically, and that in our body we might process them, we might react to them differently. So just this idea that we might react to different stressors differently, and that we might be able to track biologically what that difference is. So we have these new measures of unfair treatment and everyday discrimination. So Jackie then took that model where she combined ancestry mapping, regular association mapping, and now she added in, one by one, these measures of unfair treatment. So UT all was if anybody answered yes to any of those questions. We didn't care if it was self, spouse, whatever, just they said yes. Then UT self was if they said yes, and then when we circled back, they said it was myself. UT other was if they said yes, and then when the interviewer circled back, it was one of those other categories, anything other than self. And then the final one was the everyday discrimination value. So using these 4 models in which a single measure of unfair treatment or everyday discrimination is added, she found a number of SNPs that significantly associated with blood pressure in each case. So now how to visualize this. So she used a Venn diagram because we were interested in what kind of overlap there was in the SNPs that were identified using these 4 different measures of unfair treatment. And this is what the Venn diagram looks like. So we've got systolic blood pressure over here, systolic and diastolic, and then we've got our 4 measures of unfair treatment: UT all, everyday discrimination, UT other, and UT self. And so what we see first is that all of the SNPs between UT all and UT self are shared. There aren't any unique SNPs coming up in either of those measures of unfair treatment. So you could interpret this as saying that UT self is capturing everything that's in UT all and vice versa. Whereas then if we look at the measures of everyday discrimination and UT other, they're all unique. There's no shared SNPs with those measures. And similar things seen with diastolic blood pressure. So this then gets in with that idea that I foreshadowed, that there might actually be different ways in which these stressors are processed in the body. So again, talking to Darlene Curtis from a psychological perspective, you could think of the UT self as more direct threats, UT other as more indirect threats, or using psychology terminology, UT self a more internal locus of control, UT other a more external locus of control. So we're just we're still at the stage of trying to figure out what this means. To me, it's pretty interesting that we've got this set of unique SNPs and set of shared SNPs with these 4 measures of unfair treatment that a lot of people might argue aren't really that different. And yet we're picking up different SNPs significantly associated with blood pressure, so we've got to try and figure out what that means. So the next thing Jackie did was to look for interactions, because what I'm really interested in is the idea that the SNPs might be interacting with the sociocultural data, in this case the measures of unfair treatment, in a way that significantly contributes to the model from a statistical perspective, but also in a way that would be missed if you were a geneticist doing a straight genetics study, or if you were a cultural anthropologist doing a straight cultural study, that you can't control for stuff that you haven't measured for. So these interactive effects. So she took the same model and then she just added in a term that looked for interactive effects between each SNP that was significantly associated and then whatever measure of unfair treatment was in that model. And then this is the list of SNPs that she found to be statistically significantly included in the model and then interactive with these 4 measures of unfair treatment. So then she did the Venn diagram again as a way to visualize it. And again, so now we've got more unique SNPs. That's one difference when we're looking at the interactive effects. There aren't nearly so many shared SNPs between UT all and UT self. So we've got many more unique SNPs. Still by far the largest number of unique SNPs associated with this UT other measure of unfair treatment, which, like I said before, is a measure that nobody else has ever collected. Nobody else has ever collected this information on unfair treatment to people close to the study participant. So what do all these SNPs mean? Right? What do they do? What genes? What biological pathways? So we started to look for this. So we did a pathway analysis where we took each measure of unfair treatment. We looked at the SNPs that were interactively associated with that measure, and then looked to see what genes and what pathways those SNPs were involved in. So this is called an enrichment analysis. So you look at, it's dead. So you look at the biological pathways, and that's what this shows. So we've got signal transducer activity, circulatory system process, transport, enzyme binding, plasma membrane. That doesn't speak to me. We've got p-values here, but then we've got to correct the p-value because we're not doing a genome-wide study. We've got a custom-designed chip that has a very non-random, specifically chosen set of genes on it. So what Chris then did was correct. So we're looking to see if the SNPs that are significantly interacting with this measure of unfair treatment are enriched for those biological pathways relative to the other genes on the chip. And so what you see in this case is that all 5 of the 5 pathways that were originally identified, 4 remain significantly enriched once we correct for the genes on our chip. So this is our measure of unfair treatment to self. Now if we look at unfair treatment to all, we've got even more pathways associated, all of them significant even after correction for the genes on the chip. Now we look at UT other, so interestingly here we've got fewer pathways, even though we had by far the most SNPs uniquely associated with this measure of unfair treatment to others. And it's also the first time we see pigmentation, which is something that sort of makes sense of the pathways we might be seeing involved in this interaction between genes and unfair treatment and blood pressure. And then finally, if we look at everyday discrimination, once the pathways are corrected for the genes on the chip, there's none significantly associated. So what does all this mean? It's really kind of hard to wrap your head around all of this, and this is all pretty new stuff. Nobody's really done these analyses, so there's really no standard way of analyzing them. But what Chris decided to do was just try and look at, so this is pulling out all these different random pathways, but now let's look at all of the pathways and see where these words, these pathways fit in. So just summarizing across the 4 different types of different measures of unfair treatment, we see that signal transducer activity and transport are pathways that are identified as being enriched in 3 out of 4 of our measures. And then when Chris found, so this is some of the terms that are used to identify the genes, and more particularly the pathways associated with the SNPs that are not just on our chip, but just SNPs sort of generally. It's called annotating. If you want to know what gene or what pathway, what gene a SNP is in or what pathway a gene is in, it's called annotating. So there's certain universal terminology that gets used, and you have to figure out where you lie in this. So what we can see right off the bat is that the signal transducer activity is part of this very broad, biological processes across all of humans, implying that it's something pretty fundamental. What we don't see is stress response, because stress response, as defined at this level of pathways and annotation, is a strictly biological response. So it's like heat shock in bacteria. It's not psychosocial stress like what we're looking at. So psychosocial stress is never going to appear in this because it's not any of the terminology that's being used in the annotation. So we're still trying to figure out how to make sense of our results, which are pretty unique, in the context of what's already been published on annotating genes and different biological pathways to see where we fit in that. And I don't know if we're going to ultimately decide that this is really not the best way to visualize it, but this made me want to just step back from all this data and look at just individual genes. So the last thing I'm going to talk about is that we, again, looking for possible gene-environment interactions, again, the genetics and the sociocultural data. So from a biological perspective, G by E is defined as different genotypes that respond to environmental variation in different ways. So one of the genes that we also looked at, and this was my undergrad Alexis Bolter looked at the angiotensin 1 converting enzyme, ACE, that had already been identified and known for a long time to play a role in regulating blood pressure. And there was an allo-insertion deletion that we couldn't include on the chip because the chip could only have SNPs. So this allo-insertion deletion, Alexis just assayed on her own, and then used that same model that Jackie was using that has the genes and has all our sociocultural data, that has a measure of unfair treatment to others, and then is also looking for interactive effects. And so what she found is, in fact, a typical G by E effect. So what we see is that with both systolic and diastolic blood pressure, this blue line, which is the deletion homozygous individuals, so individuals who do not have the allo-insertion on both of their chromosomes, seem to show a unique effect of where when unfair treatment to others increases, blood pressure, both the systolic and diastolic level increases, whereas for the heterozygotes and for the individuals who have the insertion as a homozygote, their blood pressure actually decreases in response to unfair treatment. So that's what we think of as a G by E effect. It also shows that we've got this unique interaction between the genetics and the sociocultural data, here the unfair treatment going on, which is what I'm particularly interested in. And so we're trying to dig deeper into this one and see how many cases of this we have. So one of the next questions we had was, so seeing as how this unfair treatment to others measure seems to be coming up so many times, we get some of our strongest correlations with it, we get the most SNPs significantly associated with it, so who are these others? So I said that when the interviewer circled back and said, you said yes to this question, so who was it who experienced it? So it's self, spouse, parents, child, sibling, grandchild, other relative, and close friend. And then the colors here are the, have you experienced unfair treatment in your job? Have you experienced unfair treatment with the police, in the courts? So it's across all the different questions we ask. And so the thing that really jumps out at me is that we've pretty much got significant bars in all of these categories. The only one we don't is grandchild, and I think that's just because a lot of our individuals weren't grandparents. But what really strikes me is that we're getting yeses to all of this. So it means that all of these are salient questions. So unfair treatment in all of these venues and unfair treatment to all of these individuals close to our study participant are very real, very relevant aspects of this individual's life. And like I said, that has been completely missed in existing questionnaires on discrimination up till this point. So we're really digging deeper in what we think to try and figure out how exactly these experiences might translate through the genetics and into changes in blood pressure. All right, so conclusions here. So we showed that this Schreiner's approach of using both admixture and association analysis was the only one to identify a significant number of SNPs that were associated with our outcome phenotype of blood pressure. And that adding in these unique measures of unfair treatment and everyday discrimination identifies SNPs that are unique to each of these measures, suggesting that these experiences of unfair treatment and everyday discrimination may be processed differently in the body at some sort of biological level. We're particularly interested in this measure of unfair treatment to other. So, like I said, all of the relationships and categories of unfair treatment were salient. We're trying to look at it from the perspective of internal, external locus of control, direct, indirect threat, There's also possibly some sort of threshold level. So this is the same data I showed with that allo insertion deletion. It's just plotted a little bit differently so that now the lines represent different levels of unfair treatment. And what you see is that for the low levels of unfair treatment, so these darker blue lines, we're not really seeing a difference in blood pressure across the different genotypes. But that once you get up to unfair treatment measures of 3 or greater, now all of a sudden you start seeing this G by E effect where our deletion homozygotes are showing higher levels of blood pressure in response to these higher levels of unfair treatment, so that there might be some sort of threshold level of effect where at some point, the experiences of unfair treatment just become too much for the individual's body to deal with, and it manifests, at least in one way, in increased blood pressure. And then there's also this idea of missing heritability. So all of the association analyses that have been done, so we're talking hundreds of millions, billions of NIH dollars, thousands, tens of thousands of studies published looking to associate SNPs with complex phenotypes. And at the end of the day, there's only been about 5% of variation in complex phenotypes that have been associated with 5% of variation in complex phenotypes that have been associated with particular SNPs. So people are wondering, well, it's got to sum to 100. I mean, this is the variation is ultimately 100. If we're only able to explain 5% of it by this list of SNPs, what's accounting for the other 95% in variation? So it's been termed this missing heritability. Where is the effect on this other 95%? And so a lot of the discussion has focused on, well, we need more samples, we need to look at more SNPs, and that's where a lot of money and effort has gone. And to my mind, a lot of people are missing the obvious, that if you're looking at complex phenotypes, you're looking at phenotypes that are defined as having multiple genetic and multiple environmental effects, and almost certainly interacting effects between those two. But if you're controlling for one, if your geneticist controlling for the environmental stuff or vice versa, you're not going to see those interacting effects. So I really do believe that the lack of including cultural data in genetic studies and identification of interactive effects may in fact explain part of this missing heritability. And that what I'm really interested in is coming up with a set of key questions or key phenomena that can be incorporated into other association studies that possibly don't even have anything to do with blood pressure, but still capture a relevant or salient part of people's lives that impacts their health through the different complex diseases that are being studied. And I'm particularly interested in the idea of race and race-affected things like discrimination that also are being missed in a lot of these studies. So for the future, we're going to keep doing more of the same. Really try and figure out what the SNPs and pathways mean. Really dig deeper into these different unfair treatment measures. Look for gene-by-gene interactions, which I've got an undergrad working on. Lance is already looking at the personal social networks. We haven't started looking at that, but I've got a new grad student who's really interested in the idea of looking at personal social networks. So something that's been just over the past couple of years, people have started. So the idea of social networks that describe a population, this has been used for a long time to look at the spread of infectious disease. How does flu spread in a community, that sort of thing. But they found that other non-infectious diseases like obesity and happiness also seem to cluster in a network. And so on one hand, it makes sense. If you hang out with happy people who eat well and go to the gym, you're more likely to be happy and fit and this sort of stuff. But it's never really been studied, and personal social networks are a really good way to look at it. So Kia's interested in taking all of our data and throwing it at looking at depression in African Americans across the network, looking at candidate genes for depression, looking at ancestry estimates, and then seeing if we see clustering. And clustering in a network just means that there is either increased transmission or decreased transmission due to the network, due to the personal contacts of people in that network. We're also looking at telomeres as yet another measure of stress. Telomeres shorten throughout your life, but it's also thought that telomeres shorten in response to severe stressors. And then finally to add epigenetics into this study as well. So on that note, I'll thank all the members of my lab who are truly amazing. We've been very fortunate with funding, and thank you for your attention. And I'll take any questions. I guess I kind of went over, didn't I? Went into the questions session period.
We have time for a couple.
I was so interested to see that the reaction, kind of the protective reaction of close friends was even stronger than children. It was like, it just, I'm still thinking about it. I wonder what your response was.
You know, we're still taking in these data. You know, I mean, exactly, but you're right. A lot of people are mentioning experiences of close friends that is really important to them. And so we're looking at it in a number of different ways. So this is something also to look at in a network. What information is more likely to be transmitted in a network? Are you more likely to talk about friends, family, or are those experiences truly somehow more important, more salient, more stressful? I don't know, but to me the idea that, I mean, the part I keep coming back to is if it were me, and the difference between UT self and UT other, I would probably feel like, you know, well, if something happens to me, I've got control over it, I'm tough, I can handle it, but don't you mess with my baby. You know, don't you say those things to, you know, my little girl. So I mean, that would definitely stress me out more. So all of this makes perfect sense to me, but we are not nearly to the stage of understanding it, because, I mean, it's just so data rich here. We're, I mean, to be quite honest, just having some fun trying to figure out how things
I can imagine
going
that you
on.
must want to try to look at networks beyond the local community as well. That would be
Right.
a way to separate some other facts. Yeah.
Yeah, and the data were actually originally collected as personal networks, so just an individual's network, and now we're trying to use those data to leverage into a social network of the whole population, which is also relatively new stuff, because generally people are either personal network people or social network people, and we recently hired a social network person at UF, and we already had a personal network person, so we're now trying to figure out how to use these data to do everything. Anybody else?
There's something very interesting in potential over here, which is constrained severely by a philosophical problem. And that is materialism and functionalism in the theory of mind. You know, that you have, you know, the body and the mind. Are they the same? Are they different? How are they different? So when you're looking at complex, the heritability, missing heritability problem in the complex phenotype, I think there is a scope for keeping that aspect of it as a whole different area of philosophy of science, which we still don't have it addressed.
I mean, that's another thing that really struck me when I moved from genetic data to cultural data.
Yes.
You know, T's are always T's. It's not like I feel like it's a T, or, you know, I experienced it as a T, or, you know, my perception is that it's a T, right? I mean, a T is a T. And with the cultural data, it's all about, you know, with the Congo project, we always say that we measure stress experience. We never ask the moms, were you stressed by this? Right? It's literally just a checklist. Have you experienced these things? Now, they obviously have to be, what I would say, honest. They have to, you know, I mean, there's a whole discussion on what is rape. And you know, our interviewers are great at talking to the women, not just in Congo, but here. You know, and a lot of the women will go through, well, I'm still talking to the man. So it can't be rape because we're still talking. You know, and so educating with them within the context of that questionnaire to try to get them around to, you know, our definition of rape. Was it consensual? But I mean, it's a huge issue, and a lot of it has to do, you know, with how you perceive, how you communicate this experience, and then what's, you know, the same experience is stressful to one person, not stressful to another. So yeah, I mean, so I'm starting to be aware of some of these things, but I'm more aware of the fact that there's a whole lot I don't know, rather than, yeah, there's a whole lot more I would like to know.
So what's the evolutionary story about why there are genes that are susceptible to stress? You can see that obviously if we were to adapt to two different environments, that would make a lot of sense. But what about stress where there's going to be a negative effect of the stress on the expression?
Well, so I mean, it's all a hypothesis, right?
Right.
That there even are genes that
Right.
respond to the environment,
That's why I wanted to know
particularly
what your story
respond to
was.
stressors. But I mean, to my way of thinking, long term it might be maladaptive or negative, but short term it might indeed be adaptive. It depends on your time frame, the window in which you're looking. So I don't know if it's the system gone awry, or if it's just different time frames for the system, that there's a trade-off. In better adapting to this time period, you are then maladapted if you live till you're 50 or 60, but you might not live till you're 50 or 60, so better to be better adapted, in the short time frame. But this is all just conjecture. But I mean, to me, if we compare us to bacteria, right, where as we've gotten more complex, as presumably we're better able to adapt to the environment. Now, when I say better able to adapt to the environment, it may be that only a subset of us benefit from that ability to adapt to the environment, and that the others, have the downside of changing in response to the environment. The whole idea of psychiatric disorders, right? It's not an all or nothing, it's a spectrum. We define when a mental illness becomes an illness, right? Because we decide it's maladaptive in some way. But it's pretty much an arbitrary, definition where we draw the line of what's maladaptive and becomes illness. So in my mind, this whole idea of evolving a new mechanism, it would work for some people and not work for others. And there's just some kind of trade-off, and who knows where the, works for some, doesn't work for others cutoff comes. But pretty interesting. I mean, just to think how flexible, how infinitely adaptable we might be in because the genome, the mutation rate on the genome is not going to change substantially, right? It's never going to catch up to how fast the environment changes. So the idea that something else would evolve, this mechanism that presumably exists, methylation exists in a lot of organisms, but not all organisms. But methylation evolved to control gene expression, and then maybe we use that to, tweak gene expression in new and interesting ways.
So you mentioned in your project in Eastern Congo that you plan to include basically paternal samples in the future?
Maybe. We'd like to if we don't scare the dads off. If we don't get the dads trying to take their wives out of the study. We've been having long discussions about how this had happened.
So how do you plan to study exactly the paternal impact on basically the epigenetic inheritance of stress?
So the idea of getting the dads would really just be to have the genetic side of it, so that we know that methylation is to some extent genetically determined. So if we had mom's DNA and dad's DNA, we'd be able to say we've controlled for the genetic determination of methylation. So we're not really looking at methylation or stress or coping or anything in the dads. We just want DNA to do sequencing.
So do you think you're missing out on something by not including?
Well, we're certainly missing out on the paternal genetic component to regulation of methylation. To be honest, having moms and babies is unique enough. I don't know of anybody who has moms, dads, and babies in particular follow-up, but it's on the wish list.
Yeah, I was just assuming that since it's all obviously stressful for women, but it's also a very stressful place for men too, considering that. So,
Right. Yeah, so like I said, we're not even getting into the stress of dads because, if we were to do the study over again, if we could get dads involved from the beginning, then I'd be very interested. But since we don't have dads from the beginning, I think it's almost, a little late or artificial to include dads at the end. But if we get the dads really involved, And they're saying, well, why aren't you asking me all these questions that you're asking my wife? Don't you care how I'm feeling? Then we're going to start asking them those questions too. Tiffany?
This was great, and I was hoping you could speak just a little bit more explicitly to what you think or hope that the broader impacts of these projects would be, and also sort of speaking towards those of us who are or have been wary of genetic and epigenetic research that is race correlated or race associated, right? And sort of how you see your project maybe skirting around some of those critiques, or addressing those. No, or addressing those in particular, or sort of maybe providing a newer framework that we can think about, because it seems to me that what's particularly interesting about at least the Tallahassee project, if not also the Congo project, is that it's race-based discrimination conscious, as opposed to sort of racial categorization conscious, right? Which I think breaks that association a little bit differently than a lot of studies that we've seen, and I was just hoping you could maybe speak a little more explicitly to the both what you see the impacts are, but then maybe also for those of us who have had other reservations about this kind of work in the past.
Okay, so that's like a lot of questions. So I guess one thing that's drawn me into this is that there seem to be a lot of gaps, or there seems to be things that have been done maybe the way they shouldn't have been done. So my entree into more cultural anthropology has been gradual over the past 7 to 8 years, and very new stuff for me. So from the beginning of getting IRB approval, when I would get IRB approval for a study, I'd say, these are the questions I'm going to ask. Well, then for Lance, it's like, well, I don't know what questions I'm going to ask. I don't know what my study participants tell me what questions I'm going to ask. I mean, that was the first time I was just sort of blown away. Like, oh my God, how does the IRB approve this? You don't know what you're going to ask, and you want approval to go in and ask anything? And then I came around, well, of course it makes sense. If you know if you have a questionnaire before you go in the field, obviously you're only going to ask those questions. You've already assumed a priori that those are the only relevant questions, and if your study participant comes up with something else, you're going to say, no, I don't want to hear about that. And so Nikki now has sort of, my grad student Nikki has given up on academia, is still working very passionately with the Congolese, and so she lives in Cleveland, and she works with a lot of refugees and does a lot of translating for them to get different psychiatric diagnoses so they can get support. And so a lot of it is PTSD. And so she does translations for the doctors, and the people will tell her stories about what happened to them, and she'll then tell the doctors, and the doctors will say, that's not on my checklist. I'll put that in other, the box where you can write whatever you want, but nobody ever reads, and it doesn't contribute to the diagnosis. So I've just been really struck by these lists of questions, these prepared, published, validated questionnaires that lots of people go around using, and that I now, sincerely believe don't work in a lot of these contexts. People go and use them, and they get data, and they publish on them, but I mean, my experience working with a lot of these in Congo, the questions make no sense. You can translate them into Swahili, but they don't make any sense. And so I'm really drawn to that by the idea that some of these things might be mismeasured. So getting back to discrimination, Lance had a journal club one day on a published 5-minute telephone questionnaire on discrimination. I'm like, wow, you can get good data in a 5-minute telephone poll? I hate telephone polls. Whenever they call me, I just hang up. I mean, and I'm someone who actually uses these data. So if they can't get me, if they can't get my attention and my time, who on earth are they getting to answer these things? And in 5 minutes, you're going to get someone who doesn't know you from Adam to open up about what might be some intensely personal experiences and talk to you about their experience with discrimination. I mean, it just seems absurd. But it's published. People can go use this questionnaire, and they can get more data, and they can publish it, and they can get papers. So I was just really drawn to this idea that discrimination has not been very well characterized, and that Lance happened on some new things. So in his ethnographic interview, what came across was this idea of unfair treatment to others. Another one was anticipation of discrimination, that an individual might have a good 10 years free with no discrimination, but they don't know that. So whenever they go into a potential situation, an interviewer with, a white employer, or, a fancy restaurant where they think they're going to get the bad table, or, whatever, they kind of gird up, in case something happens, in the case they have to have a witty response ready, or, who knows what. And then it doesn't happen. But, they've had probably that spike in blood pressure. So this idea of anticipation of discrimination is another thing that's not been touched on in questionnaires. And then the final thing was the earliest exposure. So virtually all questionnaires ask people to recount their earliest experience with discrimination. And the idea is that your recall is going to be better for something that happened yesterday than something that happened 50 years ago. But if they're wrong, if it's the earliest experiences that are most salient, not only is it wrong, but it's insulting. Because it's like saying, well, you're not going to remember that stuff. You're not going to remember that stuff that possibly changed your life. I mean, it's really insulting. And so what Lance found was that, left to their own, people almost invariably recounted the first instance. So sometimes it'd be 50 years ago. The woman who said she stood outside waiting for the bus, and every morning the bus would pass her by, then 15 minutes, half hour later, it'd come back and get her. And she asked her parents, why can't I get on the bus when it first goes by? Well, because it's going to get the white kids, so they can get the good seats. And then, when all the white kids have seats, then the bus comes back and picks her up. And so there were lots of instances of this. And so Lance said, I wanted to slap a blood pressure cuff on them right then and there because I could see their blood pressure rising. It was still salient. I mean, this is when they learned the world's not fair, right? So I'm really drawn to this idea that there's a better way of measuring this, and that studies that get inconsistent results between discrimination and blood pressure and then are tempted to say, oh, well, there's no association. There's no effective discrimination. Again, how insulting is that? It's just been measured badly. It's just a bad study. So I get really passionate about that. And in terms of, so what else did you talk about? Fear of getting into genetics, epigenetics, into this whole race thing.
I can see everyone shuffling. I would love you to answer this, but everyone, we are officially at time, so I will let you guys leave if you're ready to leave. I don't have any short answer to that question, other than that it's been incredibly rewarding research. There is no point at which I felt like I trod into something I shouldn't have, or when I've told people, or when people have told me, why are you doing this, or you're doing it badly, or you're doing a bad thing. In fact, it's quite the opposite. It's wonderful. Well, let's thank Dr. Mulligan.
Thank you.